So you are in the laboratory of molecular genetics within the center, and what we are doing here is we are like manipulating a most interesting plant is not a crop actually, it's this little plant here.
Where's it?
So this plant is called, it happens to me all the time. It's called Nicotiana banthamiana. It's a native Australian plant, so it's like a model plant. Same way you might have heard about Arabidopsis thaliana. I don't know. It's like plants that are used as model to discover gene functions or like look at different plant pathogen interaction, this kind of thing. So we have this plant. This plant is a native Australian plant, and what is very interesting with this plant is that you can produce in this plant anything you want in a very short time. So we have projects, and Zach here is leading one of them, where we are trying to develop an antiparasitic compound with the plants to like, So Zach, if you want to come and maybe with just a short overview of what you are doing and show them an infiltration, a raw infiltration, so that's with water, but that's to show you, and then after that we will show you the results.
Yeah, sure, my pleasure. So Julia was just saying this plant, Nicotiana banthamiana, it's used around the world as a biofactory.
Sorry, what's your name?
Zach LeBlanc.
Zach.
Yeah.
Thank you, Zach.
Nice to meet you guys. So I used to actually work at a company that used this as its production system for pharmaceuticals.
Okay.
It can make pharmaceuticals as well as basically any other protein you throw at it. Right now, as Julia mentioned, we're trying to make an antiparasitic protein for the veterinary market, and we're using this plant to do that. The way we do it is we use a bacteria from nature that's actually able to infect the plant and take DNA from itself and put it into the plant. So we take advantage of this aspect and we grow up batches of that bacteria. We then infiltrate it into the plant. The bacteria transfers our genes into the plant, and then the plant produces our
Inside
drug.
the plant, yeah? Inside the plant. Wow.
Yeah. So, for example, let's say this is water right now, but let's say this was our bacteria containing the coding sequence for our gene of interest. That could be an antibody, it could be an antiparasitic protein like we're trying to produce, or another type of biologic that you'd want to make at large scale. So you take it, and this is at a small scale right now because we're using a syringe, but this can be scaled up even further if we use large vacuums and dunk the plants in. But I'll show you how we do it at small scale just to do a quick test. So, right, again, we're pretending this is the bacteria that contains our gene of interest.
We can see the bacteria.
Yeah, good. It grows real clean.
Yeah.
So, essentially what's that?
Injection before.
Yeah, so we put it right into the plant leaf. And this is just for a quick test because we're not going to get much tissue out of it.
Yeah.
But if, let's say we're just trying...
and then after that?
And you can see, if you go the other side.
Yeah, maybe I'll do the other side.
Oh,
'cause
that's fine, look, you can see the infiltration spot, so you can see. Yeah.
I could do another one here for you guys.
Okay.
Just make sure you get you can see it more on this side. So basically you just take the needle.
Can we do it by like sprayers, by sprinklers? So make.
Yeah, so we've got also different ecotypes of the plants, and some are very more receptive to spray, so we can spray them.
Okay.
So now this area on the plant, it's got our bacteria in it.
Inside,
Yeah, after a few days, so about 7 days, this spot on the plant will produce our protein of interest. We can then cut this out and then do whatever assay we want on that protein.
And so we've got plants that we've infiltrated with genes that code for a green fluorescent protein from the jellyfish, and we're going to show you what's we've infiltrated the plants on Friday, so not even 4 days ago, and we're going to show you what's the result. So you're going to have a visual.
Sorry, Jack. What the name of bacteria you inject to the plant?
Agrobacterium tumefaciens.
What?
Agrobacterium tumefaciens.
There's only one bacteria or a lot of bacteria?
So there are different subspecies that do similar things, but we generally pick one species to use. It's been disarmed, so in nature, what it'll do is if, like, let's say you have a tree and you're working next to the tree and you hurt the tree, you make like a gash in it.
Yes, yes.
This bacteria will come up from the soil and get inside that gash, and it'll make a gall. It'll make like a large rounded bit. That's the bacteria's life strategy. It does that to create food for itself and to survive. What scientists have done have taken away the genes that allows the bacteria to do that, to make that infection, and instead we just put in the genes that we want to transfer over.
It's a good bacteria, right?
Yeah.
It is
For
now.
us
Yeah.
it is.
Yeah.
So I can
you
show
want
you.
to make it mass production, yeah. After this test pass the procedure.
Sorry, say again?
Make a mass production for the developing these plants.
Yeah.
Exactly. That's the plan. And the advantage, because there are different production systems, like people use yeast, they use mammalian cell cultures, and those systems are very good and they produce very well, but they have issues with scale depending on your budget. So if you want to make a large scale factory using yeast or mammalian cells, like that's $1 billion factory, right? That's the price of it.
Amazing.
That's the.
For a plant,
Means that in our community in Bali, we use microbacterial agent, but in general, like a Bevuria, like a Metarhizium, like a Trichoderma, like
Yeah.
a Pedibacillus, like a
Yeah.
Bacillus, we use it. But we're doing by spray.
Oh, yeah.
Because we have no technology like here, but I think it's a very,
Yeah,
very
but this is applicable to this plant only. So
This
if
plant
you
only, only this plant only.
That's
So you make
kind
specific
of,
bacteria.
yeah.
Oh,
Well,
wow, that's
it's-
interesting.
Yes and no,
So
but yeah.
the plant has features that allow it to do that.
Yeah.
Julian might have mentioned that we have different varieties of this in Australia. This one comes from the desert, and its life
Desert, yeah.
strategy is basically to live fast, die young. So it grows up real quick and produces its seeds. As a result, it sort of got rid of its defense against viruses. And we think that this is one of the reasons that we can transform it so easily with this bacteria.
From this plant, what do you take, the leaf or the fruit or the flowers?
The leaf.
The leaf, yeah.
The leaf,
yeah.
You eat the leaf, yeah?
Yeah.
No, we don't eat. It's not like Aboriginal people in Australia. They use it as a nicotine, like tobacco to
Yeah,
chew.
yeah.
But we don't eat that. There is too much alkaloids, and yeah, it's gonna be toxic for you, or it's
Yeah,
gonna
yeah,
be
yeah.
hallucinogen or whatever.
Yeah.
Almost
So
like
it-
tobacco, yeah?
yeah.
Yeah, it's like if you ate a cigarette, you'd be vomiting in an hour. For sure.
Yeah, yeah.
I'll show you the
Please,
results.
thank you.
Thanks a lot.
Thank you.
No problem.
Always
Check,
remote.
check.
All right.
Sorry, make cutting.
Oh,
Thank
thanks
you.
a lot. Yeah, I'll be in touch.
Thank you.
Cheers.
Thank you very much.
Almost good bacteria.
Nice meeting you.
I need to bring that. So I don't think you will be able to all enter at the same time in the room, but maybe we can divide you in two groups. One group will go with Samantha. So Samantha is a research fellow in our group, and she's working on
Oh.
it's insect. No,
Insect,
it's
insect, yeah.
caterpillar.
Oh yeah, I see.
Caterpillar that are feeding on more than 300 crop species,
Yeah,
so
yeah.
every crops.
Mhm.
They are present everywhere around the world, so probably in Indonesia as well.
Yeah, yeah.
And so
Many.
what we are doing is we are developing technology, environmentally friendly technology, to protect the plants from these insects. So I don't know if you have any question or if you want to say a bit more. I don't know. The way we do that, well, we have different way, but mainly we are using RNA interference. So that's a mechanism where you introduce a gene targeting the insect. So we'll select a gene in the insect that's like involved in the growth and development of the insect, for example, and then we'll do RNA interference. So we are going to silence the gene in the insect by using some innovative technology in the plants. And we've got, as I said to your colleague before, we've got transgenic plants that already have very good effect. And now we are wanting to avoid the transgenic because it's not very well received and it's very difficult to get a transgenic plant on the market. And so we are developing spray, topical application of our technology to protect against the insects. So we try leading this research. Yeah.
Okay.
And so, yeah, I don't know if you have any question. We also have work on fruit flies. So we also develop technology against fruit fly, mainly in citrus. Yeah. And we have also like project with Art Innovation Australia. So mainly to like produce tree crops that flower faster, give more fruit, have a better productivity. Yeah. So that's kind of what we are doing. We also because these plants that I showed you before has been collected in a place that's very, very harsh climate, so it's desertic environment, and we've got ecotype of the plants that are collected in more.
Visualize this green fluorescent protein. We need a UV lamp.
Okay.
So the UV can be harmful for your eyes if you look directly into it. So this protects your eyes.
Okay.
Yeah, everything else will be fine.
Okay. So here we go.
If someone can come here and switch off the lights.
Okay. Wow.
Yeah. So come on in.
Okay.
So here
That's
I have the result of the infiltration that Julia just and Zach just demonstrated for you. So you can see this is the non-infiltrated plant. Under the UV light, it looks completely red, right?
Okay.
And then the infiltrated plants, yeah. So that's where So you see the green patches, and also on the one in the back, it's even stronger.
That's
Beautiful. Yeah, so that means that this part was the one that we were able to infiltrate the bacteria, and then now after 4 days, if you want, we can just collect this tissue and we can even extract the protein again. So,
So what is this on the bacteria in here in the leaf?
what do you mean? Sorry.
The use this bacteria in the leaf, and the green one
Yeah.
will be used for.
Yeah. So that's just a model for to test things. So we use this a reporter system. So it's visual, obviously, so we can see. So when we want to test something different, we use first the green fluorescent protein, because then we can actually see if it works well. And you can see here it works really well. And then, for instance, if you want to change something for what Zach was saying, you just replace this protein for whatever other protein you would like to see. And then you can, cool. Yes.
That's so cool.
It's a really nice experiment to do, because you can. It's really visual. If you can take photos,
Where
you
it's
can.
working on that.
And you can quantify. So, for instance, you see this in this plant and this plant, they have different amounts, right? And because those are two different
Like a
plasmid, so you can say, for instance, this one worked better because it had
more green.
Yes. So you can quantify these and you can see the difference. So many different experiments can be done using this as a reporter system.
Wow.
So yes, that's what I wanted to show you. Yeah. Do
Okay.
you have any questions? Do you want to switch on the lights? Oh, sure. That's completely right. Thank you.
Okay.
Do you have any questions or anything?
How long is it for you to think that this is ready to be put out with the growers or the farmers?
Oh, it takes a really long time,
Okay.
unfortunately. Because of course there's a lot of background research that was already done for many of these things, so you don't come out of nowhere, right?
Yeah.
But then maybe you select a different gene and then you test. That's a small scale test, as Zach was saying. Then probably from here you would.
Terima kasih.
Can we take picture?
So.
This
Yeah.
So I'm Dr. Sudipta Dargomit and Dr. Dina. So we are working basically on the plant avocado and the citrus. Avocado is a very profitable business for Australia. It produces it's basically domestically consumed here, but it has also very vast export potential. But there are challenges associated with that, like avocado requires a lot of labor, and also the water requirement is very high. So in our project, basically in avocado, we are trying to deal if we can make the avocado from a very tall one to a dwarf one and reduce the labor cost associated with that. That can increase the profitability of Australian avocado industry. So we are trying to do that with our latest technology on genome editing, where we are manipulating the genetic blueprint of the avocado genome, and this is through the technology called CRISPR-Cas9 system. Lina and myself, we are involved to create a mutation which can bring a particular trait change mainly related to the tree architecture to make them from tall to short. So there are a lot of proof of concepts going on. We are doing in range of crops like tobacco and other things before bringing them to the avocado. So what we are doing is a test of protocol. What you see the red one, we have successfully done this avocado transformation system and the genome editing system where we have already mutated them, but what we need is to grow them to the bigger plant.
Okay.
Yeah, and this is QUT is the only university who have in Australia mainly, but we know of we have developed an efficient genome editing platform for the avocado and also for the citrus. So this is the citrus. For the citrus we have
What kind of avocado? What variety of avocado? This has avocado?
we have Hass, Reed, Velvet, Kid.
Wow.
So Kid is a Australian comparatively dwarf variety. It's an Australian Kid. It's a bit challenging to do the research, but we are on track of it to make it work. But other three varieties, Reed, Hass, and Velvet, are doing well.
But the top avocado in Australia is has avocado, right?
Hass is most
Popular.
yeah. And there's a lot of potential to export that. So that's why all this R&D is going on, so that in future
Wilayahnya dari pucil.
Yeah.
And then from there we make it competent for transforming, so that the desired trait can be incorporated in them.
Okay.
So this is through genome editing. So first thing is like the molecular machinery, how to make the change, where to make the change, target genes which we want to, and then through tissue culture techniques, get the plants out of from there.
Wow.
So the plants that will be coming out will be edited having that particular trait.
How long from you take the seed until we
Yeah, so at this stage, if you say this is coming to up to 6 months time,
6 months time.
by 1 year's time, you will have a plant of that size.
Ay,
6
sabar
months
niya.
and 1 year.
Ay, sabar.
Total 1 year's time, you should have the plant like that.
But
This is the wild type, non-transformed. So this is the first we have to prove that we can multiply well, get the plants from the embryos up to this stage, and then transform it. So basically we are doing both. So with the molecular machinery and non-molecular machinery, normal plants.
Yeah.
Normal from the cell lines, so many tissue culture cells come in, and from each of those cells forming this one. So if you see this one, this is just from just a tiny one, this is form a big seed-like structure, and from there you can see it becomes like this. Wow! And then they become bigger like this. So you can see this one, these are tiny.
Yeah.
So this is from kind of seed-like structure, and then from there each of them will form a plant. So what basically we do, we are transforming the cells with our molecular machinery prior to that, what I showed you there. And then we get this white mosses, and then
and when become this plant?
if you can see this one, these are all multiple cells, each of them have the potential to form one plant.
This is only one plant, right?
No, this is from one single embryo, multiple cells have come out, and all the cells have the potential to form one plant.
Wow. So can make a lot of plant, yeah?
Yeah, it can be a lot of plants. So we have optimized the system how to
Can we put it on the soil directly?
not this one, this will die off. So that's why we need this artificial root vegetation. It will not happen if you do it outside. So this particular light intensity allows you cannot grow in others. That's why in QUT we have developed the system here. We have established the system how to make them multiply well. That's why we need this efficient lab, the culture conditions and everything. It matters a lot.
From this plant?
Yeah.
How long we can put it on the soil?
So this is ready to be transported into the soil. But what if you are talking about commercialization of the thing?
Yeah.
It will take long years, like because
No, not commercial. For the
oh yeah. So in
proper research.
one to 1.5 years time,
Yeah.
your plants will be in the soil. If you are not going to GMO, like this is genome editing. If you are not going, if you want from that tiny one to have this structure in the soil, 1.5 years time is good enough to have the plant. So you can give the farmers, if they are non-GM, you can just give it distribute the after hardening them. So this is another process going for acclimatization slowly, because this is from the bottle, now it is going to be in the natural condition. So they need to be slowly adjusting, because this leaf what you see doesn't have a wax in it. So when they come to the natural condition, they will slowly develop the wax and cuticles and everything, and that become resilient to be growing well in the soil conditions. So there is a particular step after this, which we do in our growth room condition downstairs.
This is the avocado or
This is banana. So these are
banana.
Banana.
I'm not working on that, so there is another group who is working on it.
Banana.
Citrus.
This is the citrus. So this is the citrus.
Citrus.
These are the, as I told you, these are the tiny little things. And
Citrus.
Mana Indonesia? Asik ngobrolnya. Okay, Mbak Ana, my question. Untuk citrusnya, what makes the citrus different?
if you see this one,
What is special?
the liquid culture, then you plate them and you get so multiple of things can come out.
The extract,
Yeah. So these are all cellars. This is transformed already. This is transformed. We are trying with a different gene to modify a trait, and we put them so that millions of cells can come out. These are